Okay, so yes, that was cool. All right, so um, let's go to the next one. Yeah, so this was actually a cool paper. We already discussed this in a journal club in the lab. Um, yeah, and what was really interesting about this paper was essentially they set up excitatory and inhibitory network that modeled that of the larval zebrafish, and they looked at how it represented new versus familiar odors. And so again, and the background for this is that we think that the piriform cortex, like the kind of the cortex that processes olfactory information, it has excitatory connections, and it's involved in kind of the memories about olfactory memories. And so a dominant idea about that is that of how that works are these Hopfield networks or attractor networks where every new, after learning, every odor will then produce a pattern of activity that converges to a particular memory. So it's very discrete. So uh, let's say some collection of odors will go to one category, some collection will go to another category. And what they found in this work was that actually there's if you model the system like what it actually looks like, in the zebrafish, you actually get this really interesting intermediate region where it's not quite an attractor, it's not quite so discrete, but you actually kind of get distortions in the space that reflect the odors that you've learned, and the learned orders kind of get stretched out kind of farther apart. So it's not quite, they don't get stretched out to kind of single points, but they get, the space distorts a bit, the geometry of the space distorts, and that can then, presumably aid doing things like classification without kind of maybe losing the information and getting stuck in these kind of discrete network states. So I thought it was, when we read it, I thought it was a really interesting idea and a kind of a nice way to use kind of simulation to come up with really cool new ideas about representation and memory and olfactory processing. So yeah, so I've already read this, but it'll be fun to go and talk to Claire about it and see what it's all about. Very cool. Okay, there we go. It's another connectome-based paper. So sorry, what's this one called? Connectome-constrained deep mechanistic networks enable hypothesis generation and refinement. Okay, so we've got two things now. We've got the connectome aspect, and now we've also got, it sounds like we've also got the, some aspect of interpretability coming in. So let's see. Okay, cool. So we can now measure the connectivity of every neuron in a neural circuit, but we are still blind to other biological details. Okay, including the dynamical characteristics of each neuron. So its channels, maybe calcium levels, things like this. The degree to which connectivity measurements alone can inform understanding of neural computation is an open question. That's right, because dynamics are a function of connectivity, connectivity, but then also the particular characteristics of each neuron. So connectivity is not the whole story. Empirically, many neural circuit architectures can perform the same large diversity of computational tasks, right? So connectivity to function is not one-to-one. Further, circuits with the same connectivity can function differently. For these reasons, because of the different functional properties of the neurons. For these reasons, there has been considerable debate about the utility of connectomic measurements for understanding brain function. Is it possible to use measurements of neural connectivity to generate accurate predictions about how the neural circuit functions? Right, so if there's kind of two aspects of the problem, the connectivity and the functional properties of neurons, can you say anything at all? My guess would be yes. Even in the complete absence of direct measurements of neural activity from a living brain. Here we show that with measurements of only the connectivity of a biological neural network, we can predict the neural activity underlying a specific neural computation. We constructed a model neural network with the experimentally determined connectivity of 64 cell types in the motion pathways of fruit fly optic lobe with unknown parameters for the single neuron and single synapse properties of each cell type. We then optimized the values of these unknown parameters using techniques from deep learning to allow the model network to detect visual motion. Our mechanistic model generates detailed experimentally testable predictions for each neuron in the connectome. We found that the model predictions agreed with the experimental measurements of neural activity across 26 studies. Our work demonstrates a strategy for generating and testing detailed hypotheses about the mechanisms of neural circuit function from connectivity measurements. Okay, so yeah, I think this is actually something that Srini presented, this work last year at Cosyne as well, so I guess they've gone a little bit farther. And the point is, you have the connectivity information, and you're going to set up your circuit. You have the connectivity information, and you also know what the circuit does. And so you're going to, the missing piece is like the functional properties of each neuron. And so you're going to optimize those to have the circuit perform the function that you're interested in, that you think it performs. And then you're going to compare. So you have the connectome, you don't have the functional properties, but you have the function of the overall circuit. So can you essentially fill in the functional properties given those two other pieces? And I guess what they're going to show is that indeed you can. So if you have the connectivity information and you have those, what you think a circuit does, you can actually figure out the remaining piece. So what that kind of means is that maybe there is a, one-to-one, is probably too strong, now between connectome to function. At least that seems to be what's being described here. So despite the possibilities of having different, despite the possibilities of the same connectome producing different types of behavior based on functional properties, it turns out that if you know what the function is, then the functional properties are fixed. Is that, yeah, so, yeah, interesting. Okay, that'll be cool. So I'm just wondering whether that is that to be expected. So what would be the alternative? The alternative would be that so the alternative would be, and this has been shown, so here's a case where something like this wouldn't happen. So a classic example of there being redundant solutions for the same function is in the lobster stomatogastric system. It's the classic work of Eve Marder's, where the circuit is known. So you have a collection of a fixed collection of neurons, and they're interconnected, and they're performing the same function. So I believe the function is to activate the muscles of the digestive system of the lobster in a particular sequence. So it's kind of a grinder. I think there's some stones in there that kind of grind up the food, and so you want kind of cyclic behavior to occur. And so you know the connectivity, but it turns out, and you know the function, which is to get the cyclic behavior. But it turns out there is a range of, I believe, kind of different channel distributions, the channel properties that performed it. So yeah, it's not, in that system, it's not the case that connectivity and function determine uniquely the functional properties. You can actually have different functional properties, same connectivity, and same function. So it's interesting. So I think it's not necessarily going to be one-to-one. You still might, if you ran this experiment in that setting, you wouldn't get such good predictions, I believe. But again, this is, at least their point, I guess, isn't that, well, that's exactly how it works, but it does give you some hypotheses, method of generating hypotheses. You can say, well, based on what I think the function is, I think the neurons are going to have to respond this way, and maybe they don't, and then you'll be in this interesting kind of redundant situation that then you can kind of try to describe. So still useful. Great. Okay. Okay, so this is cool. So respiration coordinates the olfactory cortical code. Okay, let's see what that is about. Okay. So primary goal of systems neuroscience is to determine how information is communicated across networks of neurons. Okay. One common framework for intra-areal, so within areal, communication is phase coding, in which the phase of responses relative to ongoing neural oscillations gates the transmission of stimulus information. While phase-locked spiking activity has been identified in many brain regions, including the hippocampus and olfactory system, there's little empirical evidence that the precise phase of a neural response is read out by downstream networks. Here we investigate how the phase of inputs from the olfactory bulb to the piriform cortex impacts cortical responses. Okay, so presumably when they're coming relative to the respiration cycle. In the system, odors activate select subsets of OB glomeruli, which respond at different phases of the respiration cycle. The glomeruli project bimitral and tufted cells to PCX, piriform cortex, where individual neurons receive input from multiple glomeruli. While glomerular response phase contains odor information, how this phase code is read out in PCX is not known. So what's known, actually work from the lab I'm in now, shows that there's two types of cells in the olfactory bulb, the tufted cells and the mitral cells. And for the tufted cells, they will, when the concentration of the odor gets stronger, they just fire more. They fire early and they just fire more. Whereas the mitral cells, they will start firing earlier within the cycle. So that information of where they're firing within the respiration cycle indicates the strength of the odor. And presumably also there's kind of a more broader aspect as well, where the most excited neurons in general will respond first. So if you're in a glomerulus that's really excited by an odor, you're going to respond earlier in the sniff cycle than others. So how is the phase code read out in the PCX? So we optogenetically activate a small subset of OB glomeruli at different phases of the respiratory cycle while recording spiking responses. Okay. We revealed that PCX neurons are tuned to glomerular stimulation at specific respiration phases. Remarkably, the preferred phase of each PCX neuron was highly consistent across different glomeruli. Interesting. Okay, that's cool. So I wonder, I guess, so they think that a PCX neuron is going to be particularly tuned to, like, let's say late responses versus early responses, rather than being tuned to a particular, early responses from one glomerulus and late ones in the other. I mean, I guess that does kind of make sense, because if you want an early-late situation, then you would have to have maybe, I don't know, a large integration time to kind of capture both, and then you all of a sudden start becoming less precise. So in some ways, it's kind of like in the locus system, where you have the Kenyan cells reading out specific time points very precisely. Okay. Blocking airflow through the nostril considerably diminished phase tuning, indicating that respiratory-induced oscillatory activity is required for phase-specific PCX responses. Finally, we show that OB, mitral tufted cells, prefer stimuli arriving during early inhalation, while phase preferences in PCX tile the sniff cycle. Okay, so OB prefers stimuli arriving during early inhalation. So I guess if you're arriving late, then you would have a smaller volume of odor arriving. So yeah, I mean, I guess you want them early, because that's when most of the air volume is coming in, I would think. Experimental, and it is kind of interesting, PCX tile the sniff cycle. So experimental manipulation indicated that recurrent circuitry in PCX is required for this shift in tuning, suggesting cortical circuits filter incoming inputs in a phase-specific manner. Together, results indicate that cortex implements phase to rate transformation to facilitate efficient representation of inputs arriving through the sniff cycle. Okay, so that's pretty cool. So you can have kind of phase information in the mitral tufted cells being transformed to the rate of neurons in the PCX, which are individually sensitive to different different phases of their inputs. That'll be really cool to see. Okay. Awesome. Yeah. Okay. Cool. This is a paper from Angus's lab. Really interesting work. I've read early versions of this, so it's going to be at a workshop as well. So let's see. So learning relies on coordinated synaptic changes in recurrently connected populations of neurons. Therefore, understanding collective evolution of synaptic connectivity is a key challenge for understanding how neural circuits learn to perform complex tasks. So how do these synapses change during learning? To address this, we investigate the structure of the 3 tensor formed by stacking the connectivity matrices over trials. So now you have a kind of a 3-dimensional object where you have a kind of a weight matrix, and then you stack it along the trial axis. By fitting RNNs of varying rank to large-scale neural recordings during a motor learning task, we find that the inferred connectivity is low tensor rank. A consequence of this, so if I recall this, so you can imagine that you have this 3-dimensional, connectivity object, but you can decompose it in terms of kind of outer products, 1-dimensional outer products, in the same way that you can decompose a matrix in terms of an outer product of one vector and another as a sum of those. Here you can do the same thing for a tensor, and these guys find that the connectivity, the connectivity structure they find has low tensor rank. So you need a small number of these kind of these three outer products to capture the weights that are learned. By fitting of RNNs of varying rank, a consequence of this constraint is that neural connectivity evolves over a fixed low-dimensional subspace throughout the entire course of learning. We next validate the observation of low tensor rank learning in RNNs which are trained to solve common neuroscience tasks. finally present a set of mathematical results bounding them. Yes, this was cool. One of the things I liked about their paper particularly was that they could actually then, they did some math to show that, not that this is just kind of something that they find empirically, but that you can actually, the learning process has a bound on the tensor rank of the networks that, the connectivity that you learn. Finally present a set of mathematical results bounding the matrix. Yeah, exactly. We show that low tensor rank connectivity emerges naturally in RNNs. Taken together, our findings suggest that low tensor rank connectivity is a general property of learning in recurrent connected neural populations. So that's really cool. That's kind of become very useful to lots of folks because, you know, as I've been saying, that's the big theme of, you know, training RNNs to perform various tasks, and then you want to look at what connectivity is learned, maybe to interpret it and to understand it. And these guys have shown that, okay, well, the connectivity you learn is going to have this really useful low tensor rank property, which will make it very interpretable, you know, much easier to interpret. Based on these results, we introduced an interpretable method to reverse engineer learning-induced changes in recurrent dynamics from large-scale neural recordings. So, to reverse engineer learning-induced changes in recurrent dynamics. Cool. Yeah. So I'm not quite sure what they mean by that. I guess we'll find out. Okay. All right, and the very last one. So inference of neural activity in connectome-constrained recurrent neural networks. So lots of this kind of stuff, right? So again, inference of, so you're going to, of neural activity in connectome. So if you have a recurrent neural network, why would you have to infer the neural activity? Let's have a look. The measurement of the connectome or synaptic wiring between neurons across large brain areas. Theoretical work has long focused on linking connectivity features of large neural networks to their emerging dynamics. So we saw this already earlier when we were looking at those motifs, how they influence dynamics. Nevertheless, the extent to which connectomics data can provide insight into the functionality of neural networks remains an open question, given that the physiological properties of individual neurons are unknown. To address this, we developed a theory that describes a space of solutions when inferring activity in connectivity-constrained neural networks with unknown single neuron parameters. Okay, we study recurrent neural networks in a teacher-student setup where the teacher RNN represents a biological neural network for which a connectome is available. The student has access to this connectome, but not the teacher's single neuron parameters. We then optimized the student to match the activity of a subsample of teacher neurons. We found the activity of unrecorded neurons can be inferred well above chance given a small number of recorded neurons. The minimum number of recorded neurons depends on the dimensionality of the network dynamics, but not the number of units in the network. In contrast, when the connectome is not known, inference of unrecorded activity is unsuccessful. We further demonstrated that the connectivity constraints modify the landscape of solutions, making it consistent with high-dimensional convex optimization problem. That sounds really useful. Okay, so altogether, we characterized how measured connectivity of a network beyond a core statistical description can bridge micro and mesoscale descriptions of neural dynamics. Our theory provides a guide for when the connectome data can or cannot facilitate the inference of neural circuit function and draws attention to a qualitative difference in the solution space of connectome-constrained and unconstrained networks. That's I need to read that again. So, okay, can provide insights into the functionality of neural networks. So the extent to which connectomics data can provide insight into the functionality of neural networks remains an open question, given the physiological properties are unknown. Okay, so we developed a theory that describes a space of solutions when inferring activity in connectivity-constrained neural networks with unknown single neuron properties. We studied the recurrent neural networks in teacher student where the teacher RNN represents a biological neural network for which a connectome is available. The student has access to this connectome, but not the teacher's single neuron parameters. So you basically see, so you have an RNN, you have the connectome of the RNN, but you don't have the teacher's properties of the single neurons. We optimize the student to match the activity of a subsample of teacher neurons. So maybe you subsample the teacher RNN, it has some dynamics, you know the connectivity that's producing it. And now you want, so this is in some sense related to one of the previous ones that I think Srini stuff, where you have connectivity. There you had function, here your function just to reproduce this RNN activity. Then optimize the student to match the activity of a subsample of teacher neurons. we found that the activity of unrecorded neurons can be inferred well above chance given a small number of recurrent neurons. I see. So I think what they're saying is you optimize the activity of the student network to match the activity of a subset of the teacher network neurons, and then you say, well, what about the remaining neurons? What's the activity there? And it turns out that you can actually infer those quite well. And I think that's going to be useful, right? Because when you have the connectomic information, you might have the connectomic information, but you might not have recordings from all the neurons in the circuit for which you have the connectome. Maybe you have a kind of small population of them. And the point is, given the connectivity information and given the activity of those small numbers of neurons, can you predict the activity of, if you match the activity of the small subset of the neurons you do have, can you then, how well do you predict the rest of the activity of the rest of the system? And it turns out you can do well above chance, given a small number. How many of these you need depends on dimensional network dynamics, but not the number of units in the network. Okay, so that kind of makes sense, because again, we've seen that what usually matters is the kind of intrinsic dimension of the activity, not how many neurons are involved in it. When a connectome is not known, inference of unrecorded. Okay, so, and that's cool. We further demonstrated that connectivity constraints modify the landscape of solutions, making consistent with the high-dimensional convex optimization problem. That's really cool, and I'm very excited to see that. Characterize how measured connectivity of network beyond the core statistical description can bridge micro and mesoscale descriptions of neural dynamics. Our theory provides a guide for when connectome data can or cannot facilitate the inference of neural circuit function and draws attention to a qualitative difference in the solution spaces of connectome-constrained and unconstrained network. That's really cool, so I'm really excited to see that. I think that'll be very, very interesting and very useful for the kinds of data that are coming out of our lab now, among others. Good. Okay, so yeah, so I think that's all of it. That's all of them that look particularly interesting. And there's a lot of cool stuff happening, and these were, you have to really focus now and pick a few, and I think it's a good selection. So I will try to catch some of those at the poster sessions tonight and maybe post summary of what I learned. And it seems like in terms of the themes that I thought would show up, the transformers was just the one LLM paper, but there does seem to be a lot on connectomics, trying to explain connecting various aspects of connectomes to kind of neural activity. So that seems to be a really big theme this year. Excellent. So I think that's about it for now, and I'll give an update after the poster session.